Welcome to Proving the Point, where we discuss important research papers that you should know about. Today we're going to discuss this paper called Slow CCL2 Dependent Translocation of Biopersistent Particles from Muscle to Brain. And you wouldn't know it from the title, but this paper is a vaccine safety study. And what they mean by translocation of biopersistent particles to the brain is basically when you get a vaccine, it's typically injected into the muscle, as you know. But there are components of certain vaccines that can cause these aggregates to form in your body. That's these particles that they're referring to. And this paper shows that those can be transported to the brain by your body, and they can stay there for a long time, which is what they mean by biopersistent. So we're going to talk about what these particles are, why they're in vaccines to begin with, and what effects they might have on the body. Before we're getting into it, the reason this paper matters, it should be obvious, but vaccine safety is obviously a very controversial topic. And so most people, when they discuss vaccine safety, they don't really reference the actual research literature on the topic. They tend to reference arguments from authority. And so we're actually going to take a look at a primary research article that looked at vaccine safety. And what you need to know for this paper's background, we'll go through the methods in more detail in a minute. But what you need to know for background is that vaccines typically contain a substance referred to as an adjuvant. And what this is, it's basically a molecule that they put in vaccines to intentionally activate and stimulate your immune system. So you may know that the way a vaccine works is it contains a deactivated or a weaker form of the pathogen, like the virus or the bacteria. And your immune system recognizes that and then builds up a defense against it. Well, your immune system's only going to recognize that and become active against it if your immune system is stimulated. And so the vaccine also needs to have an immunostimulant to activate the immune system. And that's what the adjuvant is. And what most vaccines use as their adjuvant is aluminum, the metal aluminum. So most vaccines, they contain aluminum as it's injected into the muscle. The aluminum basically damages the muscle, and in damaging the body, that activates the immune system and tells it that there's a threat in the area. And then the immune cells basically come to the damaged muscle. They see that there's also the weakened virus or bacteria there, and then they mount an immune response against that, and then you have resistance against that virus or bacteria. So that's the basic idea behind vaccines. The question is, what are these adjuvants that are intentionally designed to damage your cells and activate the immune system? Are they overall harmful to health? And that's actually still a question in the research field that isn't well answered for many adjuvants. So this one, like I said, it looks at aluminum. What they find, again, without getting too ahead of ourselves, is they find that when you inject aluminum containing vaccines into mice, if you inject it into their muscle, ultimately that aluminum makes its way to the brain, where it might damage brain cells and presumably impact brain function. So we're going to discuss what they did in this paper and what results they came to. In terms of their methods, again, we'll get into more detail in a moment, but in simple terms, they basically took mice and injected them with a vaccine in their tibialis anterior, which is basically the muscle in front of their tibia, so in their shin, to mimic injection of a muscle in humans. And then they used various techniques to look to see where the aluminum from the vaccines would go in the body after injection. So did it stay in the muscle, or did it spread to other organs? Short answer is it ended up spreading to other organs. And they were able to observe that by multiple different methods. Okay, they also investigated the mechanism of how did aluminum spread to these different parts of the body. And basically what they found is that immune cells were coming to the injection site, so they were coming to the muscle where the vaccine was injected, and they were eating the aluminum, presumably trying to get rid of it, because immune cells, they basically eat dead cells and toxins and waste and debris in a site of injury. And so immune cells were basically eating these aluminum particles that were being injected, and they were then moving throughout the body and transporting this aluminum to other organs. So again, the big conclusion here is that when you inject aluminum into the muscle, it can actually make it to other organs such as the brain, where it presumably has a negative effect. So this calls into question then the true safety of many vaccines that use aluminum as an adjuvant. Okay. So in terms of the use of aluminum as an adjuvant, it's common and has been for a long time. So they say here for decades, they used aluminum as an adjuvant. Specifically, it's in the form of aluminum oxyhydroxide. Okay. And again, the reason they use this is because it serves as an effective stimulant to the immune system. So it activates your immune cells. But they say that the mechanism of how it activates the immune system is not well understood. I think it's safe to say that in general, the immune system tends to become active whenever there's an injury to a tissue. So if you're not aware, the immune system, it does more than just protect you from viruses and bacteria. The immune system is also in charge of like repairing injuries. So if you get a scratch or a bruise, or your cells in a particular tissue die, it's your immune system's job to come in and start fixing that. So your immune system is activated in response to injury. So my assumption is that the way that these adjuvants are actually activating the immune system is primarily by damaging your cells and therefore causing an injury that your immune cells then respond to. Okay, they talk about how aluminum is generally well tolerated. In other words, usually these vaccines don't cause a problem in people, at least not one that's noticeable. But in certain individuals and at certain times and in certain vaccines, aluminum-containing adjuvants have caused disease in people. So there's an entire field of research on what's called ASIA. That's an abbreviation for autoimmune syndrome induced by adjuvants. And so what that means is basically adjuvants are inducing this syndrome where the immune system is so active that it actually targets our own bodies. That's autoimmune activity. Okay, so excessive aluminum is dangerous to the body and the immune system. All right, so symptoms of this disease, this condition, include like fatigue, cognitive dysfunction, and others, muscle problems as well. And they talk about how it's been observed already that the aluminum that gets injected, it can remain at the injection site for over a decade. Okay, so it's known that this aluminum is not very well cleared, although, as they say down here, it's not known, at least not at this point, where in the body the aluminum travels, or does it just stay at the injection site? Okay, so they give a little bit more background. They say that previous studies have shown that aluminum is not cleared very well from the body. So in other words, you inject individuals with aluminum-containing vaccines, and the aluminum stays in the body for a long time. It's not cleared by the kidneys very efficiently. Okay, and then they talk about how they've also, previous studies have detected aluminum in various organs within the body, like the spleen, the liver, and the brain. Okay. So for this paper, they're curious, one, can they show this again in mice? So if they inject mice with aluminum-containing vaccines, does the aluminum persist in the mouse's body? And two, they're interested in the mechanism. So how does aluminum stay in the body, and how does it get transported to different organs like the brain or the spleen? And so what they did is they obtained an aluminum and basically prepared an aluminum-containing vaccine that was like tailored to mice, but it was adjusted in a way that would ideally mimic the doses given to humans. Okay, so again, like the specific details are not necessary for us. You can read it if you want, but basically they prepared these aluminum-containing vaccines that were meant to function in mice, They injected them into the tibialis anterior. Again, that's the muscle basically in front of the shin bone in humans and in mice. And then they looked at different organs like the muscle, the brain, spleen, to see if they could find aluminum. And so we'll talk about some of this later when we get into the results. But for example, they use a technical method called particle-induced X-ray emission to look at aluminum in different organs after injection to see where the aluminum was going. Okay, the methods get more complicated than that, but I think we'll cover those as we go into each specific result. But let's just jump into the results. So this is showing the results from figure 1. So the first thing they did is after they injected these mice with the aluminum-containing vaccines, they wanted to look for the presence of aluminum in different organs. And so this is showing basically an image of a tissue of the mice after the mouse has been basically killed. So this is, I believe this is 3 weeks after the injection of the vaccine. This is showing the muscle that was injected. And panel A is just showing the tissue itself. So each of these like circles is a cell within the muscle. But panel B is showing specifically the results of that particle-induced X-ray emission, where basically each glowing white or yellow dot is aluminum. So again, how that method works, it's more technical than we need to go into, but basically it bombards a sample with array of protons, and then that causes different elements to emit different signals. And so aluminum, they're able to detect it through this method, and this is what it looks like in their results. So all this is showing is that yes, there is aluminum still present in the muscle at 21 days, 3 weeks post-injection. And then they looked in the spleen, because if you remember from the introduction, one of the previous organs where aluminum has been found after injection is the spleen. And so again, C is just showing a tissue section of the spleen in general. If you zoom in and then do that particle-induced emission method, you again see that, yes, there are spots of aluminum distributed throughout the tissue, where the more yellow the dot, the higher the concentration of aluminum at that position. So you can see it's sort of distributed throughout the tissue, but definitely in these certain locations. Okay, and then this over here, this panel is just a more zoomed in version of this. And then lastly, they did the brain. So again, previous studies have suggested that aluminum could be traveled to the brain after injection. Here they show that. So again, this first grayscale panel is just showing the tissue itself under the microscope. And then this panel is once again that particle emission method, where this yellow region is signifying extensive aluminum present. Okay. And they look to see how long these aluminum spots persist. And they found that they persist even up to a year. Okay. So basically the higher, with this bar graph, the higher it is, the more aluminum is present at that time point. So this is day 21. That's 3 weeks after injection. And this is day 180. So basically like 6 months after injection. And this is day 365. And this is showing basically amount of aluminum in the brain specifically. Okay, so the point is here, it persists for up to a year according to this result. So right away, this is probably one of the most important figures of the paper. They're basically just showing that aluminum is indeed making its way to the brain and the spleens and other organs of these mice after injection. It doesn't just stay in the muscle. Okay, and before we get too far into it, it's important to point out that aluminum in the brain is well established to be harmful to brain function. In other words, it's long been known that aluminum is a neurotoxin. So if you just to move away from that for a second. If you were to go to Google Scholar, which basically sorts search results for only academic papers, and just type in like neurotoxicity of aluminum or something general like that, and if you scroll along, you see there's an endless number of papers discussing how aluminum is neurotoxic. So if, as this current paper seems to suggest, aluminum is making its way into the brain, that's a serious problem because, again, aluminum in the brain is known to be toxic to neurons, the cells of the brain. So a very significant finding that aluminum from vaccines is making its way to the brain in these animals. Okay, so now we're going to get into some of the more complicated figures. So honestly, this is sort of the most important figure, I would say. Aluminum's in the brain and aluminum is neurotoxic. Therefore, this questions whether or not there are potential neurotoxic effects of most vaccines. All right, but they want to be more detailed than that in their analysis. And so what they start doing now is analyzing how exactly aluminum, which was injected into the muscle, made its way to the brain of these animals. And so in order to understand what comes next, you need to know a little bit more about the immune system. And basically, when your immune cells are being trained or training other immune cells, so in other words, when there's some sort of immune activating stimulus, the way that your immune system across the body is made aware of this threat has to do with lymph nodes. So your immune cells, let's say one of your immune cells finds like a bacterial cell that's not supposed to be in the body. It will travel to lymph nodes, which are distributed throughout the body. And at the lymph node, there are many other immune cells waiting to basically be activated by a threat. So you can think of the lymph node as like a meeting point for many immune cells throughout the body, okay? And also the lymph nodes help drain out waste and excess fluid from different parts of the body. Long story short, the reason this becomes relevant is because these authors believe that it is through the lymph nodes and lymphatic vessels that aluminum is traveling. The basic idea is that after you inject the muscle with these aluminum-containing vaccines, immune cells eat those aluminum particles. They go into your lymph vessels. They travel throughout the body to various lymph nodes, and ultimately they even travel up into the brain and into other organs. You can think of the lymphatic system in this context as almost like a highway where the aluminum is being transported by immune cells. So we're going to skip through some of these figures and cover it only briefly just because, one, they go into much greater detail than we need to understand the point. And two, there's almost a lot of redundancy in this paper. They almost, show their result in a way that's overkill, showing the same result many different times. Okay, but what they're showing in panel A of this figure is basically the movement of aluminum through a lymphatic vessel. So the red is aluminum particles moving along a lymphatic vessel. This is under the microscope. Okay. And I should clarify. They switch from looking at aluminum specifically to looking at this proxy for aluminum called FLBs, that stands for fluorescent latex beads. In other words, it's kind of difficult technically to measure aluminum levels in different positions of the body and to analyze it under the microscope. Okay, it's a challenge for scientists to do that. And so rather than study the mechanism of how aluminum is moving through the body using actual aluminum, they instead use this FLB, these fluorescent latex beads, which are very easy to look at under the microscope because, as the name suggests, they're fluorescent. And they mimic many of the properties of aluminum in terms of their size of these beads, mimics aluminum aggregates that are known to form in the body. So it's a caveat with the rest of this paper is that rather than using aluminum specifically, they're using these fluorescent latex beads as a proxy for aluminum. Okay, so it's not a perfect proxy. There are certainly chemical differences between these fluorescent latex beads and actual aluminum. So take all of this with a grain of salt, but anytime you see FLB in these future figures that we're about to discuss, really that's just their proxy, their imperfect proxy for aluminum. Okay, so what this is really showing is FLB, these fluorescent beads being transported through the lymph node or lymphatic vessels, but the suggestion is that that is also how the aluminum would be transported through the body. Okay, we're going to skip some of this. Panel C, this is flow cytometry results. What they're showing here, again, we don't really need to worry too much about the details, but this is showing ultimately that it's a certain cell type that is picking up these beads, and it's a specific type of immune cell. Okay, but there's many different types of immune cells. We're not going to go into the details there, but they're trying to figure out which specific type of immune cell might be picking up these beads, aka basically aluminum, and transporting it throughout the body. So we'll skip some of this. What this figure is showing is basically at different days post-injection along the x-axis, in the spleen, how many cells contain these beads. So in other words, now, again, instead of injecting aluminum into these mice, they're doing a separate experiment where they're injecting these beads, which are meant to indicate aluminum or serve as a proxy for aluminum. And what they find is that in the spleen, cells are taking up these beads and becoming more prevalent up until about 3 weeks after injection. And then, at the next time point, 90 days after injection, the number of cells that contain these fluorescent latex beads is lower, suggesting that, they do accumulate, but they do eventually get sort of eliminated from the body. Okay. See, it's showing basically the same thing in cells in the blood. So again, these fluorescent latex beads, they're being taken up by cells in the blood, and that increases up until day 21 post-injection, and then it starts to decline. This is basically showing in draining lymph nodes. So in lymph nodes, how prevalent are these FLB-loaded cells, fluorescent latex beads? And basically, it's just showing that, a lot of them do indeed have these fluorescent latex beads present in them. We can skip some of this. It's just showing under the microscope which cells have these beads in them. So like this green is outlining the membrane of the cell, a specific type of immune cell. And then the red is indicating the FLB. So this cell has within it one of these fluorescent latex beads. So again, if you assume that aluminum particles follow a similar principle to these fluorescent latex beads, that provides further evidence that these particular immune cells take up aluminum particles too. Okay, and this is basically showing the same thing, but now in the brain. So again, focus on the black bar. Okay, we're not really going to get into the white bar. They did some very complicated genetic modification experiments of the mice. But if you look at the black bar, these are normal mice. C57, that's a line of just genetically normal mice. Okay, and then if you look at various days post-injection, these fluorescent latex beads slowly accumulate in the brain. But then, if you look by day 180 in the genetically normal mice, they seem to be mostly cleared out. Okay, and then this is just showing cross sections of the brain and where at different days, according to this key down in the corner, these fluorescent latex beads are found. So that just gives you an idea of where they're traveling through the brain at different time points post-injection. similar. This panel here, B, is showing something similar. It's showing the actual microscopy results of these fluorescent beads in the brain at, I think this is 3 weeks post-injection. So okay, with this figure, again, not to get more detailed than we need to be, they're looking to see which cell types within the brain have these fluorescent latex beads in them after injection. In other words, again, if you assume the fluorescent latex beads is a good proxy for these aluminum particles from vaccines, then this would be like which cell types in the brain does the aluminum from vaccines go to? And the answer is, for this one, it's showing one in an F480 positive cell. That just means this is like a macrophage, which is a type of immune cell in the brain. Okay, these are showing a bead inside of an astrocyte, which is a type of glial cell in the brain, which is a type of cell that basically takes care of your main neurons, your brain cells. It helps clean them out, helps provide nutrition for them and things like that, to put it simply. This is showing a bead inside of an actual neuron, so like the main cells of the brain that actually transmit information and process information. A bead is present there, suggesting, these particles are making it directly into our neurons inside of our brain cells, which would be disruptive. Okay, and basically there are various, this is like a cell type that produces the covering on your brain inside of your skull, like a protective layer. The point is from this figure is just that these particles, fluorescent latex beads, aka proxy for aluminum, are making it into most cell types within the brain, suggesting they could cause widespread disruption of brain function. Okay, we're skipping some of this. What this is showing, skipping panel A, but looking at panel B, if you destroy the draining lymph nodes, then what happens is there are basically fewer cells that contain these FLBs getting to these different organs. In other words, if you block the lymph nodes in these mice, not as many fluorescent latex beads make it to the brain. So like 40% compared to what it would be with the draining lymph nodes. Only 40% of that value make it to the brain whenever you ablate the draining lymph nodes, meaning this is evidence that it is indeed the lymphatic system that is how aluminum is moving throughout the body to different organs. Okay, I think we can skip most of the rest of this. They use another genetically modified mouse line to see how the process might be different in people with certain genetic predispositions and problems, but it's not super relevant to our overall purpose. Same with this panel. They did this entire figure, really. They did another complicated experiment involving genetically modified mice to further prove their point, but again, it's not worth going into that level of detail for us. So the obvious problem with this paper is that up until this point is that only in figure 1 did they use actual aluminum. After that, they started using these fluorescent latex beads as a proxy for aluminum. But the obvious problem with that is they're not the same. A fluorescent latex bead is not aluminum. So there's questions about whether or not any of those results apply to aluminum. And so then they did one third set of experiments. So they had the aluminum experiment from the beginning, the fluorescent latex bead experiment, that was the last few figures, and now they're doing aluminum again, except in order to make it easier to visualize, they're basically linking the aluminum to what's called rhodamine, which is basically a fluorescent molecule. So the purpose of this is to make the aluminum easy to view in the cells, because again, that first figure, the method necessary to do that is very technically challenging and not applicable in every experimental situation. So by linking aluminum to rhodamine, you basically make the aluminum fluorescent, which makes it easier to see in the body under the microscope. So the benefits of this is that it's once again aluminum. The caveat is now the aluminum is linked to this other fluorescent molecule, rhodamine. And there's questions then about whether or not linking it to rhodamine impacts its movement through the body. But the point is, sometimes in science there's not a perfect experimental model. So you have to just understand that they're doing the best they can with the techniques we have available. So, again, there's a little caveat there. You could take it with a grain of salt, but now we're back to looking at aluminum. It's just conjugated to this rhodamine molecule. And they basically just show all the same things that they showed on the previous few graphs, which is, this is the spleen, and it's showing number of cells in the spleen that contain this aluminum conjugated to rhodamine after, I think this is, it's at various time points after injection. So now you inject the mice with this aluminum rhodamine conjugate and look at how many cells in the spleen have taken that up. And again, it rises steadily until 3 weeks post-injection, and then it falls by day 90. Okay, if you look in the brain, again, this aluminum, it's being taken up slowly by day 4. 3 weeks, it's significantly higher. By 90 days, it's even higher than that. And that's the basic idea. Down here, again, they start to look at which specific cell types might be responsible for bringing this aluminum to different locations in the body, but it's basically just a specific type of immune cell that we, again, don't need to go into. Okay, so skipping ahead. Down here, again, this is not really that relevant to the main conclusions of the paper for us, but they basically make another experimental modification where they get rid of the molecule that tells immune cells to come to a tissue. So in other words, there's this molecule, CCL2, that the body uses that as a signaling molecule to tell immune cells, hey, come to this tissue because there's an injury here you need to resolve. So if the scientists eliminated this molecule from the mice, which is what they did here, basically that caused a massive reduction in the movement of aluminum throughout the body, further suggesting that it's immune cells carrying it. Because if you eliminate the signal that tells immune cells to to go to a specific area, the aluminum levels drop off. Okay, that's basically all this figure is showing. And then there's one more figure, which is also pretty important. And this is basically just showing that, this is more just confirmation that aluminum is getting into the brain. But panel C is very important because what it's showing here, this is an image of a neuron, so a cell in the brain. And these little red dots are, again, the aluminum particles. And it's green because they've applied a stain that stains this protein IL-1 beta green. Okay, why does that matter? Well, interleukin-1 beta, IL-1 beta, it's a major pro-inflammatory gene or protein. So the fact that this cell turned very green means it's basically undergoing a lot of inflammation, which is bad for neurons. Inflammation in the brain is very toxic. Again, we could go to Google Scholar and just type in inflammation in the brain and cognitive dysfunction. And again, you can find there's literally over 1 million papers talking about inflammation in the brain and how it's a problem. So this sort of ties in the real world significance again, and this is the last figure of the paper. But basically, if we were to summarize this paper quickly, what they found is they could inject aluminum-containing vaccines into these mice in their muscle. It would transport aluminum to the brain. That aluminum would get inside of our neurons, our brain cells, where it would cause inflammation, which is destructive to neuron function. Okay, so then again, the reason this paper matters, that's the end of the paper. It should be obvious, but again, there's a rampant debate about whether or not vaccines are perfectly safe. And the answer is, well, a lot of them use aluminum as an adjuvant. Adjuvants are basically intentionally causing small-scale damage to activate the immune system. And it appears that this aluminum doesn't really get eliminated from the body very efficiently, and instead it can travel to different organs where it continuously causes damage. That's the idea. Okay, so does this mean that like vaccines are a net negative? I think it depends on like what claim you're trying to make. If you're claiming that, the aluminum caused by a vaccine is worse than the health consequences of the infectious diseases it prevents, probably not. Vaccines are generally, again, it depends on the vaccine, but a lot of them are very efficient at preventing their infectious disease. But we do have to be aware that, one, there are other ways besides vaccines to allow yourself to be more resistant to infectious diseases. Maybe we'll talk about some of those in future videos, but like lifestyle interventions. So, the benefit of the vaccine of preventing the disease, there are other things that could also do that without the side effects. And in terms of do vaccines have side effects, the answer is, of course they do. And we're still learning about what some of those side effects may be. So I'll probably talk more about vaccines and cover some other papers in the future. But thank you for listening.